hear from Mackenzie Blaile. She has worked at Children's Hospital Colorado for 2 years as a genetic counselor. Her areas of interest include neurology, neurogenetics, and precision medicine. Additionally, she works in the inpatient setting and assists with SMA newborn screen coordination. She was a presenter at the 2019 Cure SMA conference and is giving several presentations for MDA. In her free time, she enjoys exploring beautiful Colorado with her dog. So thanks, Mackenzie, for being here. I will have you go ahead and present your slides.
All right, thanks so much for having me.
Mm-hmm.
Try to pull this up and let me know if it looks okay. Thanks so much for having me. Today I'm going to talk to everyone about genetics and Duchenne, specifically. I'm sure you've heard a little bit about genetics already today, so I'm really just going to focus on Duchenne and how it's inherited through the family and the genetic cause of Duchenne. I have no disclosures today. So I'll define what a genetic condition is, go over some basic genetics, discuss specifically the genetics of Duchenne, inheritance, and review some genetic testing considerations. So as many of you know, Duchenne Muscular Dystrophy is what we call a genetic condition, meaning that it's a health condition that's caused by changes in our DNA. And overall, the symptoms of a genetic condition are related to the function of the gene. And so they're caused by the gene not working properly, typically. DMD or Duchenne is part of a group of conditions that are now referred to as the dystrophinopathies. And this is a spectrum of conditions ranging from cardiac involvement in carrier females to Becker muscular dystrophy to Duchenne muscular dystrophy and kind of everything in between. And as many of you probably know, the gene associated with Duchenne muscular dystrophy is called DMD. So I'll try to refer to the gene as DMD and the condition as Duchenne going forward. So quick overview of basic genetics. So our bodies are made up of millions and millions of cells. And within each cell is our DNA, our genetic information. Our DNA is packaged up into larger packages called chromosomes, which we'll talk a bit more about when we talk about inheritance or Duchenne. And then within the chromosomes, there are many, many genes. And so I kind of think of our DNA like a recipe for our body. So our body reads it and knows what to make, when to make it, where to put it. And it's spelled out in letters, which we'll talk about a little bit more later. Our body reads this recipe to make structural units called proteins. So proteins are just basically anything that has a function in our body. So these can be enzymes, small molecules, but I'll just refer to them all as proteins going forward. And there are many thousands of proteins that go in to make up each cell. And this is true for our muscle cells. And those are the main cells we're concerned about when we're talking about Duchenne. So here on the left is a picture of our chromosomes. They're numbered from 1 to 22, and then the 23rd pair is the sex chromosomes. Our genes are all located on specific chromosomes within each person, and they're all located on the same chromosome. And DMD specifically is located on one of the sex chromosomes, so it's located on the X chromosome. And that's a picture of kind of what the X chromosome looks like. So on the left is a male. He has one X chromosome and one Y chromosome. On the right, you'll see a female. Females have 2 X chromosomes. And the sex chromosomes are what determine sex at birth. Some important points about DMD, the gene specifically, beyond that it's located on the X chromosome, is that it's a huge gene. It's one of the biggest genes that we know about right now. And this is important to know because the larger the gene, the more susceptible it is to changes. So deletions, duplications, spelling changes, just because of its size. And the most common mutations or changes within DMD that cause Duchenne are typically deletions or duplications. So that accounts for about 65 to 80% of Duchenne and Becker diagnoses. There can also be spelling changes within the gene that cause the gene not to be read correctly by the body, and those account for a smaller proportion. This is a schematic of the DMD gene itself. So like I mentioned, it's a very, very large gene. It has 79 units called exons. So all genes are made up of units called exons and introns. Exons are the part of the gene that the body actually reads to make a protein. And the introns are what connect the exons to each other. And sometimes introns also have instructions for the body on how to actually read the gene. So this is a schematic of the 79 exons of DMD. And you all have probably heard of exons and introns before because there are a lot of new genetic therapies that are targeting exons and how the DMD gene is structured. So DMD, the gene, codes for a protein called dystrophin. And dystrophin is a very, very important part of our muscle cells. So the muscle cells within our skeletal muscle, so the muscle that attaches our bones together and our muscles to our bones. And then it's also very important in our cardiac muscle, so the muscle that surrounds our heart. And the interesting or unique feature of muscle cells is that they have to be very strong, but they also have to be elastic. So they have to be able to contract and release. And dystrophin plays a very, very important role in that feature of muscle cells. So I'll talk a little bit more about the inheritance of Duchenne, and I'll give a couple of scenarios. And it's important to remember that DMD is located on the X chromosome. So the only chromosomes I'm going to be talking from here on out are going to be the sex chromosomes. And sometimes you'll hear DMD referred to as a X-linked recessive condition, and I'll talk a little bit about why I don't call it that in a little while. I call it an X-linked condition. So that's kind of why these slides are titled this way. So the first scenario I'm going to walk through is if a mom is a DMD carrier, so she has a copy of DMD that isn't working correctly, and then a father who is not affected. So the mother has 2 X chromosomes, one of them is typical, and then the other X chromosome has a changed copy of DMD on it that's indicated by those stripes, so that X chromosome on the right. And then the father, we know, has an X and a Y chromosome, a typical X. So there are a couple of different scenarios, and I'll walk through each one. But the first one we know is that mom has to pass on an X chromosome. So she can either pass on her normal working X chromosome or the X chromosome that has the changed copy of DMD on it. So those are those 4 options there. And we know that dad either passes on his typical X chromosome or a Y chromosome. So if he passes on an X chromosome, the child will be female at birth. If he passes on a Y chromosome, the child will be male at birth. And there's 4 different outcomes. So on the very left, that child is an unaffected girl. So she has 2 normal copies of DMD. To the right of that is an unaffected boy. So a boy with a typical X chromosome, typical copy of DMD and Y chromosome. And then there's a chance that the child is a carrier girl. So a girl with 1 working copy of the X chromosome or DMD and then 1 non-working copy of DMD. And then the final scenario is that you can have a boy who is affected with DMD. And each of these scenarios has a 25% chance of occurring. The next scenario I'll talk through is that there's a mother who is not a carrier of DMD. So she has 2 typical X chromosomes, 2 working copies of DMD, and a father who has Duchenne. So he has a non-working copy of DMD and a Y chromosome. So we know the mother passes on a normal working copy of DMD. The dad can either pass on his X chromosome with a non-working copy of DMD, in which case the female or the girl children would be carriers, or he can pass on his Y chromosome, which we know would not cause Duchenne. And each of these scenarios has a 50% chance. And I think the key point here is that boys or fathers with DMD cannot pass on Duchenne to their male children. And quite frequently in Duchenne, there are people who, where the mother is not a carrier of Duchenne, the father does not have Duchenne, but they have a child with Duchenne. And this is because the DMD gene, like I mentioned, is huge. And so it's really susceptible to changes. So there are a lot of cases of what we call de novo inheritance or new Duchenne diagnoses in a child when neither parent carries a changed copy of DMD. And it's so frequent that it's actually about 1/3 or 33% of all Duchenne diagnoses are not inherited from a parent, so they're new in a child. So you've probably seen in the past couple of slides that I've put the word carrier in quotation marks. And that's because the more we learn about genetics and genetic conditions, and especially Duchenne, the more we realize that a lot of females who are, quote, carriers of Duchenne actually do have symptoms. So they might have cardiac symptoms or mild muscle symptoms. And these carriers who have symptoms are what we call manifesting carriers. And this is partially because of a phenomenon called X chromosome inactivation. So in females, when females are a couple of cells big when they're developing as an embryo, all the cells go through a process called X inactivation. So females typically don't need both copies of the X chromosome. They only need one. And so the body or the cells will randomly turn off one of those X chromosomes. And so some of the normal X chromosomes will be working, and some of the X chromosomes that have a changed or non-working copy of DMD on that will be turned on, and it's completely random. So females who have more of the non-working copy of DMD or that X chromosome turned on will typically, what we think, have more symptoms of the condition. And so males only have one X chromosome, and that's always the one that's turned on in their bodies, whereas females can look something like we call mosaic. So they have populations of cells that have the non-working copy of DMD, and then populations of cells that have a non-working copy of DMD turned on. So some people wonder, I know my son or I'm a carrier for DMD, why would I do genetic testing? And there are a lot more reasons to do genetic testing now than there was in the past. So the first reason is that we know that certain changes or mutations in the gene DMD can cause either more or less severe symptoms. It can predict what type of symptoms someone will have. It can sometimes predict the severity or progression of DMD within the patient. And as I'm sure you've heard about today or about to hear about in this talk after me, there are many, many new therapies either out or coming up for Duchenne, and a lot of them require that the patient has their genotype information or they know what kind of mutation they have in DMD. And that's because a lot of these therapies are targeted specifically at the copy of DMD that's not working in the body. It's also helpful to know what the genetic diagnosis is in the family, because this can clarify the risk for other family members to either be carriers for Duchenne or for them to have children who are affected. And I would encourage everyone, if you have not met with a genetic counselor, to speak to the genetic counselor who's in your MDA clinic or your muscle clinic. If you don't know your child's genotype, to get that genetic testing, or to talk about the risk for other family members. All right, so now I will leave some time for questions.
Thanks, Mackenzie. All right. We have a question. And I don't know if you can see that or not in your question box, but I'm going to read it. It's a little bit long. But they said they have 2 boys with 8 to 25 out of frame deletion, and my wife is being treated for cardiomyopathy, and she is a carrier of Duchenne. We want to plan for life after us, and we were thinking about having a 3rd baby. They're wanting to know if there are any avenues to ensure fertilization with his Y and her good X. We're looking at a son, and also, would there be any surrogacy options for them, or what would you recommend?
Yeah, that's a great question. So reproductive options for families where the mom's a carrier. So there are a lot of reproductive options now. If you are thinking about going down the IVF route, you can actually test the embryos before they're implanted in the mom to ensure that the embryos aren't affected. You can also find out the sex of the embryos before they're implanted. The other option is to get a spontaneous pregnancy, and there are prenatal genetic testing options that can be done between 10 weeks and 20 weeks of gestation. So they can do procedures to test the cells of the fetus.
Okay, so there's some options. I have heard people talk about, too, the inconclusive test result. What would you recommend someone do if they get an inconclusive test result?
Yeah, so that can go a lot of different routes. So there are 3 different kinds of genetic test results for when you're testing for Duchenne. There's a positive result, meaning that you have a change in DMD that we know causes Duchenne. There's a negative test result, meaning that the gene looks typical. And then there's this gray area result called a variant of uncertain significance, meaning that there's a lane change, a deletion, a duplication within the gene, but we're not sure what it means yet. In those scenarios, your team will, or your doctors will typically recommend some other types of testing. So before we knew the genetics of Duchenne, there were other types of testing that we would do to diagnose it. So we knew about Duchenne before we knew the gene for it. And that could be things like muscle biopsies, CK levels, other kinds of biomarker tests that we could do to either confirm or rule out that diagnosis. If you get an inconclusive result, your team may also suggest broader genetic testing to see if there's another diagnosis that could be explaining the symptoms.
Okay. All right. Thank you for explaining that. And then Tushar just came back. He says surrogacy options for older couples.
Yeah. So surrogacy usually can be done in a number of different ways. It can be used with the sperm from the father and the egg from the biological mother, and then those are used to create an embryo. The surrogate goes through an IVF cycle, and so you can test those embryos before they are implanted in the surrogate. If you wanted to use either a donor egg or the egg of the surrogate, then that would essentially ensure that the baby is not going to be affected. And that's because the surrogate would presumably go through some kind of carrier screening, or the donor egg would go through carrier screening. And so those eggs would not have a changed copy of DND and would be unaffected regardless of gender.
Okay, perfect. That answers his question. I am not seeing anything else that has popped up. I just want to make sure I'm not missing anything in the chat. So I think that's it, Mackenzie. Thank you so much for your time today.